Alrighty, hello everybody. Today we're going to be covering Lecture 7. I apologize if you can hear the purifier in the background. It's a little bit loud, but just bear with me. Hopefully it's not too overbearing. But yeah, today we're going to be covering DNA polymerases. There's a lot of info in this lecture, but I don't think all of it's that important, so I'm going to just try and go through what I think is important for your exam, and we'll go from there. So just a real quick recap of what you guys did last time. You guys did some supercoiling linking numbers, the topoisomerase types, type 1, type 2. If you want to look at, you want to do more review on that, go back to my previous lecture video. I've been trying to go through and see if I've made any mistakes. I've caught one so far, but I haven't caught anything else yet. But if you catch any mistakes, please let me know so I can post a comment and correct it. Yeah. You guys know that as supercoiling increases, DNA gets more compact, so we going this way, right? So we start with this, then we end up with this, much more compact. The linking number is just going to be the total addition of the writhe plus the twist. And I would say this is probably the most important equation, this one right here. The rest of these don't really matter too much. Maybe for your note card if you want to, but I wouldn't be too worried about it. For topoisomerases, we have type 1 and type 2. Type 1 cuts one of the two strands for twist relief, and type 2 cuts both strands. Type 2 also uses ATP. Type 1 is for twist relief, and type 2 is for writhe relief. Type 2 also uses ATP, while type 1 does not, and they both use the covalent phosphotyrosyl intermediate, where like a tyrosine amino acid binds to one of the phosphate of a nucleotide to do the cleavage reaction. Anyways, on to the actual content that we care about. So we're gonna start with DNA polymerases and their substrate reactions and stuff like that. So, jeez, my voice is already going, and I've literally been talking for 5 minutes max. Not even 5 minutes, 2. Okay, so, DNA polymerases. You guys know all about these things from Bio 200. I won't bother trying to explain the entirety of what it does, but, long story short, it is responsible for replication of DNA, and similar to like an RNA polymerase or anything like that, it produces DNA fragments in the 5' to 3' direction. So, yeah, and it also produces the complementary strand to whatever template strand you're looking at. So, the substrates for a DNA polymerase would be a template strand, right? It needs to read the template strand so that it can actually figure out what bases to use. It's going to be making the nascent strand. It's also going to need DNTPs to make the nascent strand. So, this forward reaction of forming these DNTPs and forming these DNA molecules is favored because, one, you're cleaving this, and also after you release the pyrophosphate, you cleave this to release 30 kilojoules. So that's one thing to do, or that's two things actually. So, like I said before, the first thing is the phosphodiester bond is more stable than this phosphoanhydride bond. So, forming a bond like this with this phosphate connected to two sugars is more stable than this bond right here where we're connecting a phosphate and a phosphate with an O. That's less stable than this. This is more stable. This phosphate to double sugar is more stable than this phosphoanhydride bond. And so that's one reason it's favorable. The second reason, like I said before, is pyrophosphatase, the cleavage of this pyrophosphate to release 30 kilojoules. That's very stable. And the third thing is the stabilizing interactions for the Watson-Crick H bonds. So these H bonds, the fact that we have these H bonds stabilizes that version, and so that makes it a more favorable product to form. Okay, so for phosphodiester bond formation, this is kind of like a little schematic of what the actual mechanism looks like. So, in the actual enzyme itself, DNA polymerase, look at that bottom right here, carboxylate groups of two aspartate residues coordinate with two metal ions. So, there's two magnesiums. These two pluses are magnesium ions, and this is that aspartate. This aspartate and this aspartate. There's two aspartates. So one of the magnesiums is responsible for deprotonating the 3' OH. This is the 3' OH. So basically, you want to turn this into a good nucleophile so that this can attack this phosphate. That's like it's drawn here with this arrow. So you have to deprotonate that. This magnesium is responsible for deprotonating that. So that magnesium is an important cofactor here. It's going to remove that H, and then that H, the OH, is going to attack as a result. So one magnesium deprotonates 3' to OH to facilitate the nucleophilic attack. That makes sense. Check that off. For the second thing, the second magnesium is important for stabilizing the incoming NTP. So you can see here, right, this 2+ magnesium ion. This whole thing right here is the nucleotide, right? Or not, sorry, wait, this whole thing coming in is the nucleotide. So you can see here for the second magnesium, it's actually stabilizing the 2 phosphates here that will eventually be released when you cleave, when this OH attacks here. So it helps stabilize the substrate, like, you know, these plus charges with these minus charges. So there's an ionic interaction there. So that's the second thing. That's what the second magnesium is for. It's for stabilizing the base so that when it comes in, it's able to stay there. And the main idea here is that, you know, the metal ions are really the ones doing all the chemistry. The R groups of the amino acids actually don't do much in this. The protein doesn't really do much. So if you remove the magnesium ions, good luck with your DNA polymerases. That would be really bad. So this is a really confusing image. All of this to say, this is just a way of showing what it looks like in actual space. So you have a template strand, you have the nascent strand, so the strand that you're actually forming. Here's the incoming DNTP. You see how the magnesium's right? This magnesium is helping to stabilize these phosphates here. This magnesium here is actually going to be used to pull off one of these OHs right here. This OH here, that 3' OH, and then you're going to use the OH that you got from here to attack here, basically. But I wouldn't worry too much about this image. As long as you understood what I was talking about up here, in this section, you're good to go for this. So, here is crystal structures. I wouldn't bother looking at these crystal structures. They're a little bit intimidating. Look at the cartoon. So we have, this is polymerase 1, DNA polymerase 1. I think it's pol 1. Oh, sorry, it's a DNA polymerase. I don't know why he's put that there. Either way, this is a DNA polymerase. There's a thing down here that shows you more about those. But so this is what you get for 3' to 5' exonucleases. Yeah, so all DNA polymerases that you can see here, right? All of these pol A, except for these two. These two don't have it, but so a lot of these polymerases have the 3' to 5' exonuclease. And this is basically like a hand. If you wanted to treat it like your hand, basically the way it works is there's an open form and a closed form. And in the open form, you know, you basically grab a DNTP, and then when you're ready to actually input that DNTP into the growing strand, you actually close the hand. Right? And then the nice thing about the exonuclease activity here is that, for whatever reason, right, if you accidentally use the wrong base here, so let's say that you grab a base, right, and you go into the closed form, like this form here. If you, for whatever reason, the base doesn't fit properly, you can use this 3' to 5' exonuclease to remove this base, and then the whole thing can reset again. Right, so essentially, if we have a DNTP that comes in, maybe it's the wrong DNTP, the hand isn't going to identify that until it hits the exonuclease site, basically. So the hand's going to close with this incorrect DNTP. It's going to attempt to form this bond here, but this bond between these two bases is not going to be very strong, and so the exonuclease is just going to go in and cleave it. I hope that makes sense. It's basically just a protective measure to make sure that mutations aren't often got, like, you know, you don't get much mutations in your DNA. This is just a nice table that shows you different aspects of DNA polymerases. So DNA polymerase 3 is the one that's actually used for replication. DNA polymerase 1 is used to remove any primers, which are RNA primers. And I can't remember, DNA polymerase 2, I believe, is for repair. It's like repairing a broken DNA. But you can see here the lifetimes and all that stuff of these different polymerases. So notice how they all three of these have the 3' to 5' exonuclease, but not all of them have a 5' to 3' exonuclease. That's specifically for DNA polymerase 1, which makes sense because DNA polymerase 1 actually like goes in from the 5' side and actually replaces the RNA molecules going from 5' to 3'. Because remember, the primer when you're doing DNA replication is actually made of RNA molecules or NTPs. So it's made of NTPs, not necessarily made of RNA. It's made of NTPs. So now if we look at the rest of these, though, they all don't have this 5' to 3' exonuclease, so it's not really necessary for them. The last thing that you can look at here is like the these numbers here. I wouldn't worry about this number too much. Don't think it's that important. I can't even remember what it means really. But I would say this is what's more important, is recognizing that DNA polymerase 3 lives so much longer than DNA polymerase 2 or DNA polymerase 1, as you can see by the numbers. I mean, the numbers aren't even close, right? It's not even close to how much longer DNA polymerase lives. DNA polymerase 3 lives, at least. Okay. This is a very simple slide. All you have to know, right, is that there are different chain terminators. A chain terminator is basically a nucleotide that stops the growth of a DNA chain or DNA or a nucleic acid. So, an example of this is a dideoxy triphosphate, a DDNTP. So a DNTP, right, is when you remove the 2' OH. A DDNTP is when you remove both the 2' and 3' OH. That's what's shown here. That is a DDNTP. Why is it a chain terminator, you ask? Well, remember, in order for DNA polymerase to continue polymerizing DNA, I need a 3' hydroxyl group, because this 3' hydroxyl group attacks this phosphate and allows us to kick things off. So you can imagine that if I don't have an OH here, there's going to be no nucleophile. If this is just the H, right, and no O, I can't attack this with an H. H is not a nucleophile, so that doesn't work. So that's why it's a chain terminator. It stops the polymerization completely. It's also irreversible inhibition. Like, you can't, once it's done, it's done. You can't go back. This is just a nice little slide to show you that, you know, chain terminators like acyclovir are actually used as antiviral drugs. And, you know, acyclovir is actually a chain terminator, and basically what's going on is it's a substrate for this viral thymidine kinase that only shows up in viral organisms, and you basically put it in. If the organism is viral, or you have this viral, if the organism is viral and the cells are viral, they'll have this enzyme present. This enzyme will phosphorylate this guy, and then it'll get turned into ACVTP, and then this will get incorporated into DNA. But because there's no like, what's it called? There's no 3' OH or anything like that. It's actually never gonna continue the chain. So it just completely stops the chain in its tracks, and it'll never go anywhere from there. You can also notice that this structure is very similar to, I believe it's like guanosine. Right? So, yeah. But the main idea of this is that because there's no 3' OH on this structure here, once it's bound into the DNA, it's never gonna work. The DNA is not going to continue growing, and this works really well in virally infected cells, because virally infected cells have this HSV-TK, while other like normal cells don't. So it's targeted specifically for those viral cells. Okay, so DNA replication is highly accurate. I mean, this makes sense, right? I mean, you wouldn't expect your DNA polymerases to make a lot of mistakes, because if they did, then we'd have a lot of differences compared to our, like, the parents and stuff like that, we'd have significantly more differences. But, you know, a lot of the DNA we have from our parents is pretty much the same, like 50% from dad, 50% from mom. If there were more error rates during this replication process, you know, maybe your cells would have different DNA, right? Not all of your cells would be made of that 50% of mom, 50% of dad. Maybe there would be some subsect of that DNA that's actually mutated after you've gone through a lot of mitosis rounds, right? If you had a lot of DNA errors, a lot of errors that could happen during replication. But there are very few errors during replication, so all that to say that, you know, it's very important that there's not very many errors, because the more errors there are, the more, you know, likely you are to get some sort of a health problem, health complication, some sort of mutation that could be detrimental for life. So, to put it in perspective, it's like the error rate is like only one typo in 1,000 books. So there's very few errors. So, the active site of DNA polymerase is actually very, very interesting. It actually, first of all, it actually will not bind normal NTPs. And the reason is because it is very highly specific for this shape of the ribose. Specifically, this OH on the 2' is too big to fit in the active site, and therefore it will not fit into the active site of a DNA polymerase. It's just too large, it will not work. So there's amino acid side chains that interact with that pentose and discriminate against that 2' OH. And the other thing is that there's one active site, right, that fits the geometry of the Watson-Crick base pairs. So any other base pairs that occur are not going to happen because the active site cannot fit them in there. Basically, once it has locked onto a base and it's looking for a complementary base, there's a very specific shape that it's looking for. And so it won't bind anything else it will be very... The chance of you binding a base that you don't want is very low, basically. Because of the fact that the shape doesn't permit you binding something that doesn't fit. So here's a kind of a diagram for how all this stuff works. So if we start from this here, let's go the correct route. So we have our C, right? If we have our C and we're going the correct direction, we're gonna add our DGTP, and it's gonna go in there, it's gonna attach, gonna go here, it's gonna go here, and then we're done, and we're gonna go to the next thing. But if we end up going the incorrect way, so let's say that instead of binding a G here, we actually end up binding an A, right? So we have an A in our DNA polymerase. Remember in that open conformation of the hand? Then what's gonna happen is the pole 1 is going to basically try and close, and it's going to get stuck in that conformation. It's just not going to work. But this mismatched 3' terminus, right? There's two options that could happen. One, it could get stuck there and actually could maybe be forced to bind, and in that case it'll move on to the next cycle. Or the more probable option, because it's so much faster, is that we will switch to the exonuclease site, remove that A, and then we'll release it, and then we'll continue on, and then we'll go back in the correct direction. So the important thing to recognize is these slow steps, right? So it's kinetically controlled, if you want to think about it. So when it's jammed, right, it's jammed in the open form, and then it's slowly going to push down and try and attach that A into the C. That isn't going to work, and the two options you have now are, okay, well, we've tried to match the A to the C. We can either move on to the next cycle, or we can switch out and remove it. And switching out to the exonuclease is much faster than allowing this to continue to try and wait for the next DNTP. So what ends up happening is you're far more likely to end up with this exonuclease activity than you are to actually get that A stuck next to that C and move in order to move on to this next DNTP cycle. All this to say that, you know, because this step is so slow, this step often dominates here when you've added an incorrect base because it's so much faster. So this is just you're not going to be tested on this, but basically what happened is the mitochondrial DNA polymerase broke, and as a result, they were screwed. This rat suffered a lot. They didn't have a lot of ATP, they had a lot of reactive oxygen species, they had a change in redox, decreased oxidative phosphorylation. They had a rough time. So alright. The next thing we can look at is DNA sequencing. I'm not going to bother with this, all this to say that DNA sequencing has gotten a lot cheaper and a lot faster over the years. PCR. You guys have already done PCR, I think, probably a billion times. Just to give like a very brief overview of how it works. If you're given a DNA strand, right, what happens is you're going to start by separating the DNA strand. You're going to do it by melting it. Generally, that happens at around, I would say, 90 degrees Celsius, something like that. So at 90 degrees Celsius, you're going to separate the DNA strands. Then, what you're going to do is you're going to do the annealing strand. You're going to take primers that you have designed, you're going to attach them, anneal them at around 60 degrees Celsius. You're going to drop the temperature. You're going to anneal them so that they get stuck complementarily to the DNA strand that you're interested in. And then you're going to add DNA polymerases, or you're going to add your DNA polymerases and your DNTPs, and you're going to elongate. And so you're going to basically form the complementary strand for the section of DNA that you attach the primers to. And then what you're going to do is you're going to do that cycle over and over again, that melting, annealing, elongation step. So after that elongation step, right, you've basically formed another set of double-stranded DNA, which you can then go back and melt, anneal, and then replicate again. And you can do that again and again and again until you form just a ton of products, basically. But yeah. Hopefully that makes sense. It's basically just a way of replicating sections of DNA that you want. Okay. Sanger sequencing. Sanger sequencing is basically just... It's not PCR per se, but it's very dependent on DNA polymerase for the chemistry. Essentially what's happening is you have a substrate mixture that has unlabeled nucleotides and dideoxynucleotides that are labeled. So these labeled dideoxynucleotides that have different colored dyes on them. And the ratio is usually like 10 to 1 for the unlabeled nucleotides to dideoxys, or like up to 300 to 1. You basically want a lot of unlabeled nucleotides and not too many dideoxynucleotides, because if you put too many dideoxynucleotides in there, you're basically just going to end up with no chains and no nucleic acid chains, because it's just going to keep terminating instantly because the dideoxynucleotides just keep binding first. So, let's say that you have this strand here given up top. Not that. This strand given up top. And you have your mixture full of unlabeled nucleotides, as well as these dideoxynucleotides that are labeled, right? There's some fluorescently labeled dideoxynucleotides. Let's say that the T's are red, the A's are purple, G's green, and then C's are blue, as shown here. What's going to happen is you're going to mix all of these fluorescently dyed DDNTPs in, and then you're going to mix in some normal DNTPs. And as you allow this mixture to mix with the DNA that you just used PCR on to replicate, these DDNTPs will bind complementarily to that DNA strand. And that binding, right, the binding of the DDNTPs
is going to lead to some sort of fluorescence when you track it on a gel. So for example, let's say if we're looking at the DDTs, right? Maybe the first nucleotide is going to be complementary to this A, right? So you'd bind the T. And if a dideoxy binds in that T position, done. Okay, this is now done. You're not going to extend it anymore. Maybe for this next one, right? For this 2 here, right? The next T is going to be 3 away. So you're going to bind a normal T, not a dideoxy T, a normal T, right? A normal DNTP, right? Or DTTP. And then you're going to bind a normal DATP, and then a normal DCTP. And then you're going to bind another dideoxy TTP, right? That's going to terminate the chain. And then you're going to go to the next one, right? You're going to have a T, A, C, T, G, A, C, and then you're going to bind another dideoxy T. That's going to terminate it here. And you're going to keep doing that for all of these bases, right? All of the... Basically, every single DNA strand that you put in your mixture is going to have some combination of these bases. Right? And eventually, it's going to get terminated. Right? It's just kind of random probability whether or not a DDNTP binds in place of a normal NTP. That's why you make sure that your ratio is more skewed towards normal NTPs and not DDNTPs. Because if you skewed it towards DDNTPs, all of them would be like this length. Or this length. They would be very short. But what this allows you to do is, remember that these DDNTPs will fluoresce if you put them in a gel. So after you've gathered all of these fragments, right, that have, you know, varying lengths because of the fact that you've put a certain ratio of DDNTPs to normal and DNTPs, you're going to put it in a gel, and you're going to run the gel. And remember that shorter fragments travel to the bottom, right? This is a gel electrophoresis, by the way. Shorter fragments travel to the bottom. So if we look at this, you can see that this is the shortest fragment. It's 1 DDNTP. It's going to be here. And it's going to shine red. Cool. The second one is this one here. It's going to shine purple because A is the second base. Purple. Blue is next because it's 3. Red because it's 4. Right? Green. And you can see how this is going to keep going, right? 5 is green. 6 is A, right? 7 is blue, I believe. 8 is red. 9 is blue, and then 10 is green, right? You can see how as you do this, you're basically able to tell what every base is at every position. And you can also use these photo multipliers to kind of help you visualize this, but I wouldn't worry too much about this photo multiplier part. All this to say, Sanger sequencing is very helpful for determining the actual sequence of DNA that you're looking at. Right? PCR only allows you to reproduce the section of DNA you're looking at, but let's say you only know the 2 sides that flank that region that you're trying to find, so you know how to make the primers for it, but you don't know what's in the middle. So if you don't know what's in the middle, you have to find out what's in the middle. And the way you do that is through something like Sanger sequencing that allows you to, you know, put your DNA in after PCR, put a bunch of DDNTPs and normal NTPs in there, and then using this kind of gel technique, you can see, oh, okay, wait, which colors fluoresce at each position, and then you can go sequence by or base by base and say, oh, the sequence is A, C, T, whatever, right? So the important thing to remember is the ratio, right? The ratio. You need to have a ratio of more DNTPs to DDNTPs, otherwise you'll stall your Sanger sequencing early, because if you have too many DDNTPs, you just don't get any chains at all that are actually long enough to do this. I hope that makes sense. This is a bit of a complex topic. I'm always at the IC, as I've mentioned this before, but you know, if you have more questions, feel free to stop by the workshops or come to the IC. Okay, next generation sequencing. Next generation sequencing is interesting because it's not really described very well. I would say the best way to think about this is how it's drawn here. So
right here, yeah, we've got this kind of plate, right? You see this gray piece here is a plate. And we've got these little pores, right, that have these sections of DNA. And so what's going to happen is you're going to put your DNA of interest into one of these little pore, like machines, right? It's going to bind complementarily to these little, not pores, but these little flags here, if you will. And so what's then going to happen is, using this little like primer here that's attached to your actual DNA sequence, you're gonna elongate this straight up, and you're gonna do that for all of these, right? So let's say that there's many of these bound here. So there's one here, there's one here, there's one here. You know, they're everywhere, right? And what you're gonna do is you're gonna elongate them upwards. And so it's going to look like this image. And the nuance to how this is different from Sanger sequencing is the type of chain terminators you use. So in Sanger sequencing, you use irreversible chain terminators. You use DD-NTPs, so they have no 3' OH, and you use them with dyes. But this version of sequencing is different because what it uses instead are these reversible chain terminators. And they're reversible because they basically, after the NTP has bound to its complementary base, it'll fluoresce, and then after it has fluoresced, you can basically cleave off this protecting group, and it'll act as a 3' OH to then allow binding of the next nucleotide. So the main idea here is that it can accept a 3'. I guess the main thing. Sorry, I should be specific here. So this is talking about the polymerase down here. So these points are about the polymerase. So this is that specific polymerase we're talking about, and this is an example of an NTP that we would use for these next-gen sequencing things. You can see this reversible chain terminator. This is the base, and then you have this massive dye, right? And this modified polymerase is actually able to accept this dye as well as the base. So that's the important thing here. So sequencing polymerase, it's a modified DNA polymerase. It can accept the 3' azetyl-methyl group, so this group here. It can also accept the dye. This whole thing about accepting scars left on previous bases, I would ignore that. I don't think it's that important. The word scars, don't really know what that's supposed to imply here. But I don't think that that's super helpful to what this actually is. I don't think that's super helpful for what this is trying to get across. And the other thing is that it's very good at polymerizing. So the initial versions of this started at like 5 base pairs. You know, it didn't do a lot of base pairs. Then it went to 50, and then it went to 300. So basically, it's very efficient at what it does. And this is basically what it looks like if you do a sequencing. So basically what you're going to do is you have a camera, right? So let's say that I'm keeping track of like this one spot here, this one blue dot, right? Essentially with these reversible chain terminators, what's going on is, like I said before, right, you have these like primers you have in here. You're going to put your DNA in, right? And then you're going to elongate, right, using some sort of polymerase, like a normal polymerase. And then when you are gonna actually do the part where you're trying to find the sequence of this section of DNA, right? So you're gonna try and find what this like stick of DNA is here, right? That nucleic acid. What's gonna happen is since you have now formed this complementary strand, you can get rid of this this black piece of DNA. So that is going to disappear. It's going to wash out. So now you're just left with a bunch of the complementary strand. So what you can do is you can use these reversible chain terminators, right, that have a die on them, similar to how in Sanger sequencing they have a die, right? The only difference between Sanger sequencing and next gen is they have reversible chain terminators. So now you can go down to your little plate, right, and you can add base by base. So you can add a base here, right? And then what's going to happen is that base, you're going to take a picture as it glows, right? So you can see, like, as it glows, right? You take a picture, and it's going to be blue, okay? And then that reversible chain terminator is going to come off, and then it's going to bind the next nucleotide. That's going to glow another color. Maybe it glows red, right? So then I'm going to take another picture of that. And so maybe you'll see blue, and then you'll see red. And then you're going to do that process over and over again. So you'll see blue, red, yellow, green, whatever, right? And you're going to do that over and over again. Just keep taking pictures of that until you get the full DNA sequence. And so you may be wondering, why do I have, a million of these? The nice thing about next-gen sequencing is that I don't just have to look at one piece of DNA. I can look at thousands to hundreds of thousands to millions at once. And that's nice because that means I don't have to run it over, like run the sequencing over and over again. And so if I'm just curious about one piece of DNA, I'll just look at one specific spot and just track that spot until that section is done. If I want to look at a different piece of DNA, I'll just pick that spot and then track that spot until it's done. Or maybe this spot, I'll look at that spot instead, or this spot, right? You see what I'm saying? So you can do this for any piece of DNA. This is why it's more helpful than Sanger sequencing, because Sanger sequencing you can only do one piece of DNA at a time. But with next gen, you can do as many as you want. That's that. Actually, that's actually it for the entire lecture. But just to really quickly recap for this, I want to make it clear that for next gen sequencing, the main thing you should know is that the way it's different from Sanger sequencing is the fact that it uses a reversible chain terminator. That's the main difference. So the reversible chain terminator allows you to attach the base onto the complementary strand, and then it's going to glow. That reversible chain terminator is going to come off, and then you're going to attach the next base. That's going to glow. You're going to take a picture. It's going to come off. Attach the next one. Glow. Take a picture. Come off. Right? You see what I'm saying? So, like, you're going to attach the base. The terminator is going to get removed. Or, sorry. Attach the base. Attach the chain terminator. The thing will glow. You take a photo. Then you remove the chain terminator. And then you attach the next base. That next base is going to glow. And then you're going to take a picture of it. You're going to remove the chain terminator. And then you're going to add the next base. And you're going to keep repeating that process, right? You're going to attach the next base. It's going to glow. You're going to remove the chain terminator. Attach the next base. Or take a photo as well, of course. Attach the next base. When you attach the next base, it's going to glow. You're going to take a picture of it. You're going to remove the chain terminator. Attach the next base. So it's just that process over and over and over again. But other than that, this is just a nice little image of how the cost of next-gen sequencing has gone down over the years. But yeah, other than that, that's it for this video. I hope it was helpful, and if you have questions, feel free to, leave a comment or stop by the IC. But yeah, have a good one, guys, and happy studying.